So the last video that you may have watched was talking about how the electric part of the message, the electrochemical message, travels along the neuron. This part is going to highlight what happens between the neurons. Okay, so the chemical part of the message. And it was covered through some questions that were handed out in the electrochemical message little handout that we gave out. Okay, so it's the last slide. We have a look at it. The first question was to take this diagram and label all of these things on it. Okay, and then you want to be able to use it to do number 2. So explain what happens when the wave of depolarization, so the electric part of the message, reaches the end of a presynaptic neuron. Alright, so the first part was to actually label this. So since it takes quite a bit to try and label with pen, I'm going to show you it labeled and explain how you figure out some of the labels. Alright, so the first thing that you should look for is to see where is the action potential. Okay, where is that depolarized region that's traveling along the neuron? Because that's one of the things that you had to label. Okay, so if it's depolarized, remember the charge has reversed. So normally in a resting membrane, it's positive on the outside and negative on the inside. But where the message is at that given time, it's going to be depolarized, so the opposite. So negative on the outside and positive on the inside. So we know we can label this part right here, the depolarized regions. Hoping that you can see, but I might take a pen maybe. Okay, so right here, this is the depolarized region. Okay, so ahead of it, so we know that the message traveling down the neuron always goes from the dendrite end towards the axon terminal. So it will go in this direction, as indicated by the arrow. Okay, so it goes in this way, heading towards the axon terminal. So that's the electro part of the message, the electric part. Okay, so ahead of that wave of depolarization, the neuron would be resting, waiting for the message to come along it. Okay, so right ahead of it, this region here would be resting. So it's positive on the outside, negative on the inside, and the ions are on the right sides. Okay, so lots of sodium on the outside and potassium on the inside. Right behind it, right behind the depolarized region, the charges are back to normal. Okay, so you can see it's positive on the outside, negative on the inside, like it would be if it was resting, but the ions are mixed up. Okay, it's still got potassium on the outside and sodium on the inside. So we call this region right behind the message the repolarized region. And it will quickly undergo a refractory period where the sodium-potassium pump will kick in and get the ions back to normal. But right behind the depolarized region, it is considered repolarized. Okay, so that's part of why the message isn't going to go backwards, it's going to keep going forwards. Okay, so the positive influx of sodium kind of instigates the next resting area to start depolarizing. Alright, so the electric part of the message is moving down towards the axon terminal. When it gets to the end, it kind of bumps into this channel. So I'm hoping this is highlighting here, but this channel right here, this calcium channel. So I should label it calcium channel, and it's showing a bunch of calcium ions outside of the neuron. Okay, so when the electric message reaches the calcium channel, it opens it and it allows the calcium to come in. So you can see the green arrow showing the calcium coming in. And the calcium will bind to these little structures here, these little sacs. And we learned in 521 that these kinds of sacs, vesicles, okay, it's actually a synaptic vesicle, if you want to be more specific, but the vesicles are meant for transporting things. Okay, so these synaptic vesicles have neurotransmitters inside. So somewhere you should have labeled these little circular things as neurotransmitters. So here's our label up here. Okay, so there's these little sacs, the vesicles, synaptic vesicles, sitting there holding neurotransmitters in. And it depends on what kind of neuron, which neurotransmitters it has. Maybe it's dopamine, maybe it's serotonin, maybe it's glutamate, acetylcholine, this depends. Okay, so when the calcium comes and binds to the vesicle, it causes the vesicle to move, be carried towards the membrane surface. Okay, so the end of this axon terminal, and it will fuse with it. So this one's showing it's just starting to fuse here, but the one down lower, it's already fused and opened up, allowing the neurotransmitters to go out into the space. Okay, so we call this space the synapse, or the synaptic cleft, or the synaptic gap, but it's the space that's between the two neurons. Now, in terms of other labels, since this neuron, the first neuron, is before the gap, we call it the presynaptic neuron. Okay, and in particular, it's pointing to the axon of the presynaptic neuron. The one after the synapse, or after the gap, is called the postsynaptic neuron, and we know that the receiving area of a neuron is the dendrite, so it must be the dendrite of a postsynaptic neuron. Now, in order for these vesicles to be moved, okay, it takes a lot of energy. So that's why in this diagram you also see these little organelles. If you remember, we used to call them the powerhouse. That's their nickname, because they are important for helping us get as much energy out of our food as we can. So that is the mitochondria. So there's a lot of them around because we need lots of energy in order to process the message, okay, in order to carry these vesicles and allow them to fuse and release their neurotransmitters from one of the parts. Okay, so the movement of the neurotransmitters between the neurons, that is the chemical part of the message. So I'm going to write chemical. You can't really see it very well here, but that's kind of right. Let's see. Put in some text. Okay, so that's the chemical part of the message. And put an arrow there. But basically, it is what allows the message to go from neuron to neuron, because the electric part can't jump the gap. So we have neurotransmitters that carry the message to the next neuron. Now, when they go to the next neuron, they will bind to receptors. So these are receptors on the dendrite. Okay, so it's labeled here. Pen. Okay, there's the receptor. Then the receptors are usually linked to some kind of a channel. If they're linked to a sodium channel and enough sodium gets to go into that neuron, then it will excite that neuron and cause the message to be passed on. If they're linked to, say, maybe a chloride channel or some other kind of channel, then they would make the inside even more negative and prevent the message from being passed on. So it would inhibit firing of the neuron. Okay, so it depends on what's going on in your body and what kind of neurons they are, all that stuff, what kind of receptor it'll bind to, and what it will cause to happen in the next neuron. In any case, if the neurotransmitters were left out in the space, then they would be there constantly, and the message would either be inhibited all the time or stimulated all the time, and we can't have that happen. That's not how they work. Okay, it's because the message, we have to have clearing of the neurotransmitters for the next wave of polarization to have its effect. Okay, so for either turning on or turning off or intensifying the messages, you have to have clearing of the neurotransmitters. Now, there's a couple ways that happens. On this diagram, we have what we call reuptake channels. So here's one here. Okay, it's pointing to the one at the bottom here. And basically, it does what the name suggests. It takes the neurotransmitters back in, and they will get stored in vesicles again. That's one way. Another way is by having them be broken down. So there'd be enzymes, okay, little proteins out in this gap that would break down the neurotransmitters. So we're always breaking things down and recycling them. That's one of the things that could take place. So usually it's either they have reuptake or they might have enzymes to help break them down. So, let's see, I think I'm going to find a little animation.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Acetylcholine molecules diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium ion channels. This causes the ligand-gated sodium ion channels to open, and sodium ions diffuse into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
Hopefully that helped when you can actually see it moving and visualize what happens. There's a couple other animations that are really good too. This chemical synapse one. The neuromuscular junction one, you can watch that. This is showing the end of a neuron where it is meeting up with its effector. Okay, so
an action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholine esterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
So that one showed one of the ways that the neurotransmitters can be removed from the synaptic cleft before another message fires. Okay, so in that case, an enzyme was there to break it down, and parts of it were able to be recycled. Okay, so that is the chemical part of the electrochemical message. The second question asks you to actually list the steps. So if you read in your book, there's a little section in your book that kind of lists them out. This is what you could say in your 6 steps. Okay, the wave of depolarization reaches the axon terminal, triggers the calcium ion gates to open. Calcium influx, that just means calcium enters and causes the vesicles to fuse with the membrane and release their neurotransmitters into the gap. The neurotransmitters, so that's NT, neurotransmitters attach to specific receptors on the postsynaptic neuron. So they travel across the gap and they bind to the receptors on the postsynaptic neuron. And the postsynaptic neuron is going to be either excited or inhibited, depending on what happens. Okay, once they reach the threshold level, it could be excited, or if it's a different type of ion that's allowed in, then it could be inhibited. The neurotransmitter is broken down by an enzyme or taken back into the presynaptic neuron using reuptake channels. Okay, so you should be able to label and describe what happens in the electrochemical message. Question 3, oops, I think we'll use, there, said what does it mean if the postsynaptic neuron is excited or inhibited? Ooh, that's kind of small. So if it's excited, it means that they must have opened some sodium gates, and basically when enough sodium has gone in, it reaches the threshold level and it causes or triggers the next wave of depolarization to happen. Okay, so it causes the action potential to happen in the next neuron. If it's inhibited, then it makes it more negative on the inside usually, so it takes it kind of farther away from reaching its threshold to be able to have the neuron fire. So it could be that it opened a chloride channel or some other means of inhibiting it from happening. No action potential would happen. Question 4 asked how the neurotransmitters are removed from the synapse. Okay, so there were a couple ways. Either enzymes are released from the presynaptic neuron, or as we saw in the animation, they could be on the postsynaptic neuron. They could be bound to either of the membranes, but in any case, enzymes could break down the neurotransmitter, or they might be taken back in through reuptake channels. Okay. Question 5 asked you to list and describe the 6 types of neurotransmitters. So you should have an idea where you might find them and a little bit about a couple things that they might be responsible for. So the first was acetylcholine. And if you remember back to when we were learning about the PNS, acetylcholine is released at the ends of the parasympathetic nerves, but it's also released, it's the main one for the somatic nervous system. So if you can remember things that the somatic and parasympathetic nervous system look after, you could state those as some of the jobs of acetylcholine. Okay, so the parasympathetic nervous system, remember, it slows your heart rate, it slows your breathing, it constricts your airways to like normal levels, it constricts your pupils, those sorts of things. And the somatic means that looks after your voluntary movements, so acetylcholine is important at your muscles, so at the end where it meets your muscles, acetylcholine is important in muscle movements. Noradrenaline is also known as norepinephrine. That's the one that's released from the sympathetic nerves. So it's involved in the stress or fight or flight response. Okay, so if you think of all those things, your increased heart rate, increased breathing rate, opening up of your airways, dilation of your pupils, all those kind of things would be taken care of by noradrenaline being released at the ends of the sympathetic nerves. Glutamate and GABA are two of the primary neurotransmitters found in your brain. So glutamate is excitatory, so it increases the level of firing in your brain, whereas GABA is inhibitory, so it will slow down certain processes in your brain, cause them not to happen. Dopamine and serotonin are also found in your brain, and they have specific roles. Dopamine is involved in the control of your movements. Okay, so I don't know if we've covered it yet in class, but when we talk about Parkinson's, for instance, Parkinson's, there's loss of a lot of the nervous tissue that is involved in dopamine production and control of the movements. So the movements are extremely affected in people with Parkinson's, and by the time it's figured out you have Parkinson's, a lot of the destruction has already happened. Okay, so dopamine has a major role in controlling your movements, but it also has a major role in your reward pathway. So it helps to elevate mood and moderates your feeling of happiness and reward. Okay, so it's often affected by the use of drugs. Serotonin. Serotonin also affects mood. Okay, so if you have depression, for instance, depression is often associated with lower serotonin levels. Okay, it affects your mood and your sleep patterns and your ability to focus, that kind of stuff. It's often associated with your serotonin levels, and sometimes dopamine as well. But serotonin affects your mood, your sleep patterns, sleepiness, and your level of alertness or focus. Number 6. Does the nerve impulse cross the synaptic gap? Explain. And the answer is no. The information that's carried across the gap has to be carried chemically. Okay, so the actual nerve impulse, the action potential can't cross the gap. You have to have neurotransmitters kind of carry the message along. Number 7. JJ. Oops. Yes, it was there. There we go. Number 7 asks you to define or explain a couple of things. One was the all or none principle. So if you read about it in your book, basically, once the threshold of the stimulus is met, the message is going to be sent along the full length of the neuron. It's not going to go part way. There's no part message. Okay? It's like they talk about it and liken it to the trigger of a gun. Once you pull it, then if you pull it harder, it's not going to make it go any faster. But once you pull it, it's going to go. Unless there's something broken in the gun. Just like unless there's something wrong with the nerve or neuron. Threshold level, basically that's the minimum level or minimum amount of stimulation that has to occur in order to bring about depolarization of a neuron. They can be stimulated by chemicals, by heat, mechanical movement, or by neurotransmitters that are released from earlier neurons. So one neuron releases some neurotransmitters, then they can either combine their effects and bring about the threshold level in the next one, so on. Intensity. Okay, so how intense you feel a certain thing, or how the level of pain or degree of hotness, for instance. Okay, depends on the number of times the neuron fires. So the greater frequency, number of times it fires, the greater the intensity. So it's really important that those neurotransmitters are cleared out of the synaptic gap, because it allows for the intensity to be truly felt, because then each message will come across and be felt as it should. Okay, be registered as it should. So it's the frequency that dictates the intensity of what we feel. Okay, so that covers most of the chapter, except for some of the disorders, procedures that we'll be reviewing in a different way. Again, you can look at the notes about them online, and there may be some in, I can't remember if there's some in the Quizlet on them or not, but you could create a Quizlet about them. Later we'll be doing, if you haven't already done it, we'll be doing a investigation on how different drugs work and how they affect the neurons, and how they can interfere with the regular impulses, and how they're received, and what normally happens in our brain and nervous system. So if you have any questions or need some help with any of this, please don't hesitate to ask. That's what I'm here for.